Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will learn about the continuation of the topic, the concept of pH and buffers. In the last video we have learnt about the concept of pH. Now we will study about buffers in detail. Buffer. Buffer is a solution that resists changes in pH when small amounts of acid or base are added. Means in definition, we have seen that it resists changes in pH. Resist means it can withstand. It can withstand changes in pH. Means if there are changes in pH also, buffer can resist. When there are changes in pH, when small amounts of acid or base are added. So here buffer, it is a solution that resists, means it can withstand changes in pH when small amounts of acid or base are added. Means in simple words we can say that it keeps the pH steady or constant, which is very important in our body and even in experiments also. Let's learn about the components of buffer. A buffer usually contains a weak acid and its salt. Example, acetic acid and sodium acetate. Two, weak base and its salt. Example, ammonia and ammonium chloride. Here, these are the components of buffer, whether it is weak acid and its salt or weak base and its salt. Now let's learn about how buffers work. Here we are taking example of an acetic acid buffer. In this buffer, it contains weak acid and its salt. Here what is weak acid? Acetic acid, and salt is sodium acetate. So if you take weak acid, that is acetic acid, it is a substance that partially ionizes into acetate ions and H plus ions when dissolved in water. Means partially ionizes. Partially ionizes means it can get back to again acetic acid. Here the symbol showing reversible, means both ways it can undergo reactions. Now let's see about its salt, that is sodium acetate. So again, this is a neutral compound that produces conjugate base when dissolved in water. Let's see, CH3COO dissociates into CH3COO minus. This is a conjugate base, plus H plus when dissolved in water. Now let's see why is the salt important. Because salt provides conjugate base. So here what is the salt? we have seen in the acetic acid buffer is sodium acetate. And what is the conjugate base? Acetate ion CH3COO minus when reacts with acid means H plus. It forms acetic acid. So here conjugate base is removing extra H plus ions from the solution. Meanwhile, acetic acid reacts with hydroxide to form acetate ions and water. So here acetic acid is an acid, hydroxide ion is a base, and in this reaction acetic acid is removing extra bases from the solution. So this combination, what is this combination? The combination of weak acid and its salt help resist the changes in pH. Now let's see how pH is maintained stable by adding acid or base in buffers. What happens when you add acid? Conjugate base reacts with acid to form acetic acid. So here these extra H plus ions are removed by the conjugate base, keeping the pH stable without dropping much. 2. When you add base, acetic acid reacts with base to form acetate ions and water molecule. So here acetic acid is a acid, hydroxide ion is a base to form acetate ions and water molecule. So here hydroxide ion OH minus ion is neutralized by the acid, keeping the pH stable without rising much. So finally, by seeing this both, we have come to the conclusion that buffers can neutralize both acids or bases and keep the pH stable or constant. Let's learn some of the important points under buffers. Buffers, these are very important in biology and chemistry. In our blood, pH stays at around 7.4 because of which buffer? If you know, comment me. In our blood, pH stays at around 7.4 because of which buffer? If you know, comment me. Second, enzymes work at specific pH levels. Buffers help maintain that. And finally, in industries and labs, buffers are used in reactions, food product, and even in medicines. What will happen if buffers are not present? If buffers are not present in system, especially in biological systems, even small additions of acids or bases can cause drastic changes in pH, which can lead to serious consequences, such as first, enzyme activity will be lost or it will be affected because most of the enzymes work at specific pH. Without buffers, their activity will be lost. Second, disruption in the cell membrane takes place. So all cells maintain stable internal pH, that is homeostasis. Without buffers, cell membrane can be damaged. Third, imbalance in blood pH. Uh, our blood pH ranges from 7.35 to 7.45 because of buffers. Without buffers, these can uh, lead to changes in pH. If it is too acidic, it leads to acidosis, and if it is too basic, it leads to alkalosis. These both conditions are life threatening if they are not corrected quickly. Fourth, protein denaturation. And a sudden change in pH can drastically alter the charge of the protein, and even it leads to unfolding. So without buffers, their activity is also lost. Fifth, lab and industrial reactions will fail. So, in labs and industries, uh, especially food industries or whatever, chemical reactions all depend upon specific pH. So, here without buffers, the chemical reactions will give the unreliable results, or they stop completely reacting. Let's quickly revise what we have learned under buffers. We have studied that buffers are the ones that resist changes in pH using weak acid or weak base pairs. And we have studied even components of buffers. In that, we have taken example of acetic acid buffer. And we have also studied the importance of buffers. And finally, what will happen if buffers are not present. I hope this video help you understand the basics clearly. If you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. In the next video, we will learn the topic on weak acids and bases. Until then, keep learning and stay curious. See you in the next class.